In Chapter 15, we're going to be looking at inheritance from a chromosomal level. So the man who's primarily responsible for our understanding of chromosomal inheritance is Thomas Hunt Morgan in the early 1900s. And Thomas Hunt Morgan did not work with plants. He actually worked with fruit flies, Drosophila melanogaster. And fruit flies are those teeny tiny little flies that you see buzzing around your fruit, about the size of a match head. And they're really great model organisms for a couple of different reasons. Number one, they don't have that many chromosomes. They're really small, so you can keep like thousands of them in a drawer in the lab. They're very easy to breed. They don't really need a lot of materials. They don't eat a lot. They're not dangerous, so it's a lot better than using like a tiger, for example. So for those reasons, Drosophila melanogaster has become a staple in the genetic research community. Okay, so before we can get into any of the real details of our Morgan fly experiment, we have to talk a little bit about what a normal Drosophila melanogaster looks like and the notation that we use specifically for fruit flies. So here's my fruit fly. It has a giant compound eye. It's got a pair of antennae. It has a mouth part for eating fruit. It also has a thorax, and attached to that thorax we're going to have a pair of wings for flying, and then they also have a pair of tiny little wings called halteres that are not for flying. They're actually just for steering. We also have our legs attached to our thoracic cavity here, and then we have the abdomen. Okay, so that is our generalized fruit fly. They're usually gray. Okay, so when we're talking about fruit flies and different traits on them, we're going to assign a letter just like we used to in Chapter 14. So in this case, we have chosen the letter B, and this is going to be for body color. And a normal fruit fly is actually going to be gray. So since this is normal, this is the wild type condition, this would be analogous to the dominant condition. We would give it a superscript plus sign to show that this is the normal, dominant, wild type condition. The other allele that we're following in these experiments is going to be the wings. So this is going to be wing configuration, we'll call it. So if you have normal wings, where you have one pair of big wings and one pair of little halteres, that would be the wild type, so it would be noted as VG+. And if it's anything else, then we would just omit the plus sign from that. So our Morgan fly experiment, the first one we're going to talk about, he took a regular wild type fly that is both gray, so normal, and has the normal wing configuration. So since it's a P generation, it has to be homozygous, so we have B+, B+, and VG+, VG+. And then he crossed this fly with another one that was a double mutant. So this mutant has a black body, so it gets BB for its genotype, and it has vestigial wings. So that means that if we're looking at the thoracic cavity here, instead of having a pair of flight wings and a pair of halteres, they're going to have two pairs of halteres. So these are going to be flightless black fruit flies. So if we were going to make a Punnett square of this particular cross, okay, we know that our P generation is going to produce 4 gametes. Okay, and in those gametes, we're going to have B+, so the wild type allele in all of them, and we're also going to get VG+ in all of those gametes, okay, because that's all they have to offer. So we'd have B+, VG+, or B+, VG+. Those are your choices from our wild type fly. The double mutant is also going to give rise to 4 different gametes, and each one is going to have the mutant version of the body color, black, and the mutant wings, the vestigial wings, which they've assigned as VG. So we'll end up with B, VG, B, VG. So we fill this out, we end up with B plus B, VG plus VG, and that would be the case for all of these squares. Okay, so it will be heterozygous for each trait. And that's exactly what Thomas Hunt Morgan saw in his experiment. We have one copy of the wild type, one copy of the mutant for the body color, one copy of the wild type, and one copy of the mutant for the wing configuration. So, consequently, their phenotype is gray with normal wings. So then he took it one step further. And instead of crossing these F1 flies with each other like Mendel did, instead he's going to do a test cross where he crosses it again with a homozygous recessive for both traits. And he did this because he wanted to test out Mendel's assertion that all alleles are going to be sorted into gametes randomly. And this would imply that there's only one gene on every chromosome. So if that were the case, we're going to fill out a Punnett square and see what would happen if that were true. One gene per chromosome. So here we're looking at our papa fly. This was our double mutant. Okay, so it had the genotype BB, VG, VG. So if there's only one gene on each chromosome, that means that we're going to have two chromosomes, one for body color and one for the type of wing configuration that our fly has. Now they're diploid cells, which means we're going to have two copies of each one. Okay, so this would be our papa fly. So after the S phase and meiosis, each of our gametes would end up with one copy of each chromosome. So they get one copy of the chromosome that has the black allele, body color, and they would get another chromosome that has the wing configuration, in this case the vestigial flightless wing phenotype. Now, mama fly was the F1 generation, so mama fly is heterozygous for both traits. But again, we're still assuming that there's only one gene per chromosome. So we have one chromosome that has the mutant gene from this mama fly's dad, and then we have one chromosome that is homologous that has the wild type gene for gray body color. Same thing with their wings. It would be a different set of chromosomes. We have one that is wild type and one that is mutant. So after the S phase and meiosis, we would end up with these gametes. So we'd get one gamete that has wild gray color and normal wings. Another one has gray and vestigial wings. Another one would be black with normal wings, and the other one is black and vestigial wings. So they're all different combinations if they're on different chromosomes. So when we make our Punnett square, we have our mama fly on the top, and then we have our double mutant test cross dad fly on the side, okay? So we have all of our combinations from our mama fly and all of the combinations from our double mutant. And if we were to fill this out, we would end up with a certain ratio. So let's fill out just the top portion because all of the other rows beneath it are going to be exactly the same. So we have B plus B, VG plus VG on that one. So the next one, B plus B, VG, VG. Next one, B, B, VG plus VG, and then the last one, B, B, VG, VG. So if we were to assign a phenotype to each of those, this one would be gray, and it would have normal wings. Okay, the next one would be gray as well, and it would have vestigial wings. Okay, next one is going to be black with normal wings, and then the final one is going to be black with vestigial wings. So we end up with a 1 to 1 to 1 to 1 ratio in that F2 generation. Now there's the possibility that there are multiple genes on each chromosome. So in this hypothesis, hypothesis number 2, we've put the genes on the same chromosome. Because remember, at this point in time, we don't know, is it one gene per chromosome or are there several genes per chromosome? So we look at this and we have a new configuration. So Papa Fly here is going to have 2 copies of that one chromosome that has both genes on it. And Mama Fly also has 2 homologous copies that have both genes on it, but they're going to be different from each other. So we've got our wild type on this one and the normal wild type on the other. We've got mutant and wild type on the other. And we do not have a combination where we would have B plus VG here because Mama Fly would have inherited this chromosome here from one of its parents and the other chromosome from the other parent. Okay, so we go through the S phase, we go through meiosis, and we end up with the gametes. Okay, so gametes are going to be the same from our double mutant as they were if the genes were on different chromosomes, but we have something different happening on our gametes from our mama fly, our F1 fly. So here we have double mutant on this one, double mutant, double normal, double normal. Okay, so we do not have all combinations here. We only have double mutant or double normal. So we put that into a Punnett square again, and I really only have to fill in 2 boxes on this one, thank goodness. So we're going to have B plus B, VG plus VG, okay? And that would be the same all the way down. It would also be the same on this column, since these 2 gametes are the same. Then we have BB, VG, VG, all the way down, and same thing here, because these 2 are the same. So if we were to describe their phenotype in this scenario, we would have gray body, normal wings. Okay, same thing here, gray, normal. Okay, on the third one, we would have black, vestigial, and then the last one would also be black and vestigial wings. So that means that we are going to have a 1:1 ratio of gray normal and black vestigial. So we will have no gray vestigial or black normal. So that should not be a thing. Okay, so here we have a summation of this experiment. We have our predicted ratios right here. Okay, so if the genes are on different chromosomes, we should end up with a 1:1:1:1 ratio of each of these different scenarios. So we should get equal numbers of wild, mutant, mutant, wild, mutant, and mutant, wild. If they're on the same chromosome, so that second hypothesis that we went through, then we should end up with wild, wild, and mutant, mutant, but we should not end up with any wild, mutant, or mutant wilds. So those were our predictions. At the bottom, we have the actual results, okay? So all of the flies that he encountered, all of the F2 generation, he ended up with 965 wild, so normal, 944 mutant, 206 normal mutants, and 185 mutant normal. So it doesn't really fall into either of our hypotheses, right? But it has to be one or the other. You can't kind of be on the same chromosome and kind of not be on the same chromosome, right? Like you either are where you're not. So it has to meet one of these predicted ratios. So what we got to do is look for the one that fits best. And the one that's going to fit best is genes on the same chromosome. We have roughly equal number, 965 and 944, for double mutant and double normal. And we have almost zero of our 185 and 206, which are mutant normal and normal mutant. So how did that even happen? How did we end up with a normal mutant and a mutant normal? So it turns out that this is a result of crossing over during meiosis. Crossing over does not always happen, right? It is a random thing that happens sometimes. So if we look at our male fly here, it was homozygous for both traits. So even if crossing over were to happen, you'd never know about it. All of the sperm have exactly the same chromosomal configuration. But if we look at the female fly that was heterozygous, so we had double mutant and double normal chromosomes, during the S phase, okay, so that's here, each of those would chromosomes would replicate, so now we have two sister chromatids, right? And then during prophase 1 of meiosis, those two homologous chromosomes would line up right next to each other, and we could have crossing over happen between them. Okay, so that is the swapping of DNA, and that's what we see in this part of the drawing here. It's the physical swapping of DNA. So now we have new combinations. Now we can have our double normal, our double mutant, but since crossing over occurred, we now have a normal mutant and a mutant normal. And that is what accounts for those 200-ish weird combinations that we did not predict from our original Punnett squares. So this series of experiments by Morgan showed that there are multiple genes on a single chromosome. So there are times when you will most likely inherit two different traits together. They're not all independently assorted. There are more than one gene on each chromosome. And so genes that exist on the same chromosome are considered linked. So for example, we have the gene P, A, and B that all exist on this same chromosome, so they are linked together. Most of the time, they will be inherited together. The dominant version of P, the recessive version of A, and the dominant allele for B will all be inherited together unless crossing over happens between the homologous chromosomes. So that's the only way that we would end up with a dominant version of B, for example, with a recessive A and a dominant P. So one of Thomas Hunt Morgan's graduate students, Alfred Henry Sturtevant, took this one step further. So his main goal was to map the genes, right? So we know which genes are on the same chromosome. Now I want to know where are they on that chromosome. So to do that, he would use all of these experiments and calculate out what was the % chance that crossing over occurred. Okay, and then he would use that to make a map. So at the bottom of the slide here, we can see one of these chromosomal maps. Now keep in mind that this is the early 1900s, so we cannot sequence the genome and use computers and things like that. That didn't happen until the early 2000s. So we are 100 years before that was even possible. So on this chromosome map that we have here, we're following 3 genes. We have our body color, we have our vestigial wings, and then the middle one is cinnabar eyes. So this is going to be eye color, okay, which we haven't talked about yet, but that's what it is. And so he's arranged them such that body color and vestigial wings are farther apart from each other than cinnabar eyes would be from either one. And that is going to be based off of the math from these experiments. So if I look at my experimental results here, and I add up all of those offspring, so 965 plus 944 plus 206 plus 185, that gives me 2,300 total offspring in that F2 generation, okay? Then if I add up just the ones where crossing over occurred, it gives me 391. So 391 out of my 2,300 offspring had crossing over between homologous chromosomes during meiosis 1. So if I calculate that out, that gives me a 17% likelihood that crossing over will happen between the two genes, body color and the wings, which is what we see right here. Okay, 17%. So that means that they did a bunch of other experiments that were following body color and cinnabar eyes. And they found that in 9% of that F2 generation, there was crossing over between them. And then they had to do another experiment that was following cinnabar eyes and vestigial wings and found that there is a 9.5% chance of crossing over happening between those two traits. So if you do this enough, right, this is like millions of flies that you're breeding and figuring out which ones have crossing over, you can map out where the chromosomes or where the genes are along the chromosome. So we have these percentages, so like 17% would be 17 map units. Okay, so one map unit is the 1% likelihood that crossing over would happen between two genes. Okay, as you can see, if we compare 17% versus 9%, 9% means that they're going to be a lot closer to each other than 17%. They're farther apart. And that makes a lot of sense, right? Because if we have homologous chromosomes here, and I'm following two genes, so maybe I've got a gene here, and I've got another gene right here, okay? They're right next to each other. The likelihood that we would have crossing over happen right here is highly unlikely, right? Conversely, if we were following two genes and maybe we have one gene here and the other gene is here, the chance that crossing over happens somewhere in between them is going to be a lot higher, okay? So the higher the likelihood of crossing over, like 17, the farther away they are. The smaller the chance of crossing over, like 9, the closer they are to each other. Okay, and map units actually have a very specific unit associated with them. They are centimorgans. So this would be 17 centimorgans in honor of Sturdivant's graduate advisor, Thomas Hunt Morgan. So here we have another Morgan experiment, and in this experiment we're following eye color. So we have a female with red eyes, cross it with a male that has white eyes, and not surprisingly we get an F1 generation that all have red eyes, kind of reminiscent of our purple flower, white flower pea plant experiment. We let the F1 generation breed with each other, and the F2 generation ends up with a 3:1 ratio, just like in our pea plant. So that wasn't really that surprising. However, what was surprising was that most of the time, almost all of the time, the white eyes would be found on a male, not a female, which raised some questions for Thomas Hunt Morgan. So we have to talk a little bit about the determination of sex. So traditionally in humans and in flies, we use the XY system. So if you have two copies of XX, then you would develop as a typical female, and if you have one copy of X and one copy of Y, you would develop as a typical male. Now, of course, we talked earlier about how sometimes it's not quite that simple, but generally speaking, that is going to be the situation for both humans and for flies. That is not the case for other insects like grasshoppers. They're going to use an X nothing system. So if you have 2 copies of the X chromosome, then you're a lady. And if you have 1 copy of the X chromosome, then you develop as a male. Birds, snakes, and butterflies, the obvious pairings, right, are going to use the ZW system, which is almost opposite of what we're used to. So if you have 2 copies of Z, then you develop as a male. And if you have Z and a copy of W, then you develop as a female. And then finally, in our social insects, things like ants and bees and wasps, critters that have a pretty complex social hierarchy, they're going to be based off of diploid and haploid. So if you are diploid, then you are a female. And if you are haploid, then you are male. But back to our flies. Flies are going to use the same sex determination system that humans do, so X and Y. So here we have a female fly with red eyes, okay? So if it's a female, that means that it's going to have 2 copies of the X chromosome. So these Punnett squares get a little bit more complicated because we have to follow the chromosome in addition to the alleles, okay? And this is crossed with a male that has white eyes. Now the normal configuration is to have red eyes, right? So the wild type allele, which I'm going to assign W, would be red eyes, whereas the mutant would be white eyes, okay? So our female here, since this is the P generation, has to be homozygous, is going to be W+, W+. And our male is going to be XW. And I've assigned it to the X chromosome because this particular gene is located on the X chromosome. It is considered an X-linked gene. Okay, so let's set up our Punnett square. We've got mama fly here, which is XX, with our W+, W+, and then papa fly is going to be XW and Y. We fill in our Punnett square. And just like in chapter 14, we put the wild type first, if it's there, just by social convention. And we end up with these possible genotypes. So if there is a female, okay, so we've got a female here, it's going to have red eyes. Here we have a male with red eyes. Here we have a female with red eyes, and finally a male with, you guessed it, red eyes. So we saw exactly what he saw. We had an entire F1 generation with red eyes. Okay, so now we're going to allow this F1 generation to breed with each other. Okay, so we've got mama fly here, XX. Mama fly has all red eyes, okay? So from our Punnett square last time, we learned that mom is going to have XW+ and XW because they're going to inherit an X chromosome from dad here, who was X mutant Y. So that means that this female could only inherit this XW from dad and the XW+ from mom, okay? So that's mom of our F1. Papa fly here is going to be X and Y, and it's only going to inherit that X chromosome from the mother, okay? Y would come from the dad. So this would be XW+ and Y. So we fill this out again. And this is what we end up with. So we'll have a female with red eyes. We'll have a male with red eyes. We'll have a female with red eyes, and then a male with white eyes. And that, again, is exactly what Thomas Hunt Morgan saw in his experiments. There's a 3-to-1 ratio of red eyes to white eyes, and that white eye is male. So that experiment was really valuable because it helped us see that there are genes on our sex chromosomes, X and Y potentially, that are not related in any way to sex. Like your eye color has nothing to do with what sex you are or determining what sex you will be. So they're completely unrelated, but they're located on sex chromosomes. So we would consider them to be sex-linked genes. So you tend to inherit them with your sex. So some human sex-linked genes would be red-green color vision. So the dominant version of this allele would allow you to differentiate between red and green, whereas the recessive allele makes it a little bit more challenging, as we can see in this example at the bottom, where normal color vision, where you have a green hat, a yellow shirt, and red shoes, if you have the recessive version, or color blindness, as they might refer to it as, everything kind of looks like a version of yellow. So it's harder to differentiate between reds and greens. And that's because you just don't make those cones in your eye. Another sex-linked gene would be hair growth, where the dominant allele would be normal hair growth, and the recessive allele would be male pattern baldness. Okay, and we call it male pattern baldness, even though it's on the X chromosome, because it happens more frequently in men. Just like red-green color blindness is more frequently seen in men. And then the final one would be production of blood clotting proteins, which would be the normal allele, and then the recessive allele would give you hemophilia. So I mentioned that baldness and color blindness is going to be more common in men than it is in women. It can still happen in women, but it tends to be more common in men, and there's a reason for that. So we're going to go through a couple Punnett squares here to see what happens in these different scenarios. So in our first one, we have a female, and let's say that we're following color vision here. Who has color vision? Okay, and we have a male who is color blind. Okay, has the recessive allele. So if we were to make this Punnett square and we fill it out, let's see what would happen with their offspring. Whoops. Okay, so in this first example where we have a woman with normal color vision who has children with a colorblind male, we would end up with female daughters who have color vision and male sons who also have color vision. So everything's fine there. Let's do the next example. So in this example, we have a woman with color vision. She can differentiate between red and green. And then we have a male who also has color vision. Okay, so let's go through this Punnett square. Okay, so if we go through our predicted possibilities here, we'd have females, which are all going to have color vision, so that's fine. And then when we look at the males, we have one that has color vision and one that is now color blind. So there's a 50-50 chance of getting color vision or being colorblind. And that's because we only get the Y from dad and we get one copy of X from mom. So if mom is a carrier, like she is in this case, there is a 25% chance that they will have a son who is colorblind. Okay, let's look at the last example. We have a female who is a carrier but has color vision, and we have a male who is colorblind. Okay, so go through our Punnett square here. Whoa. Okay, so here we go. Look at our females. We've got two females on the top there. There's a 50-50 chance that they will have color vision. So one of them is going to be homozygous recessive for color blindness. The other one will be heterozygous. If we look at the males, we again have a 50-50 chance of color vision. So one of them will inherit the color vision allele from mom. The other one will inherit the colorblind allele from mom. So as you can see, it can happen in females, but it is going to be more common in males. Which brings us to our last topic of this chapter, which is X inactivation in female mammals. So as it turns out, we don't actually need 2 X chromosomes. We only need 1. So consequently, during early embryonic development, if we have 2 copies of the X chromosome, we will inactivate one of them and turn it into a bar body. Now, which one we inactivate is completely random and different in every single cell. So in the picture at the bottom here, we see that some of those cells have inactivated the orange X chromosome, and some of them have inactivated the black X chromosome. And so as a result of this, we end up with females that exhibit a mosaic pattern of that trait, like a tortoiseshell cat. So tortoiseshell cats are always female because the allele for fur color is found on the X chromosome. So in order to have both alleles, orange and black, you'd have to have 2 X chromosomes. So what ends up happening is during fertilization, the zygote inherits 1 allele for black fur, 1 allele for orange on the X chromosomes. Then during early embryonic development, those are randomly shut down in every single cell. So we end up with patches of orange fur, and in those patches, the cells have shut down the black allele. And then we also have patches of black fur, and in those cells, they have created a bar body out of the X chromosome that had the orange allele. So we end up with this mosaic pattern in the female cats, whereas males would only be black or orange. So now that we have a basic understanding of how we inherit our chromosomes using independent assortment and meiosis and how many genes are on the chromosomes, etc., we can move on to the last topic of this semester, which is gene regulation. So how do we know which genes to turn on in the cell and when?